We
Great.
go.
Okay, wonderful. Welcome everyone to the QLS seminar series. It's my pleasure to introduce Asher Leeks, who will be telling us about the social lives of viruses. Really fascinating topic. Asher recently, just almost a year ago, joined the faculty at UBC in the Department of Zoology as an assistant professor of evolutionary biology. Before that, he was a postdoc at Yale University with Paul Turner for a couple of years. And I guess he's slowly been making his way westward. Before that, being at Oxford for his undergrad and PhD in evolutionary biology, where working with Ashley Griffin and then Stu West, um, developing a lot of the ideas that I think are really coming to fruition in his independent group now. And I don't think it's an exaggeration to say that Asher is one of the leaders in this burgeoning field of social lives of viruses. So, welcome, Asher. It's a pleasure to have you and take it away. I think also Asher is happy to take questions during the seminar as well, and then after as well. So I will moderate and try to keep things on track, but raise a hand if you have a question, and if not, we'll take some questions at the end. So thank you, Asher. Take it away.
Perfect. Thanks, Jesse, for the lovely introduction and for inviting me to this great series. Um, so yeah, as Jesse said, please do interrupt with questions kind of as I go along, and this can be a little bit discursive. Um, so I'm not in the room right now, but normally when I tell people that I work on the social lives of viruses, you kind of get funny looks, right? Because we're not used to thinking about viruses as social organisms or using that kind of language. Instead, we're used to thinking about, like fluffy mammals or birds or bees or that kind of thing. And the reason is that when you open any textbook that kind of talks about viruses, the typical depiction that you'll see is of viruses as like a singular particle, right? Like a piece of bad news wrapped in a protein coat is a description, I think, from Ray Anderson. So my argument today that I'm going to try and do through a couple of different stories is I'm going to try to make the case that you looking at viruses through the lens of social evolution can be a useful way of doing so. And actually, that in many senses, viruses are not just social, but in some ways more social in an evolutionary sense than some of these more traditional types of organism. Okay, so I'm going to begin by just talking about like what the hell I even mean by viral sociality, like what are we talking about kind of physically? And then we're going to talk about two stories on kind of different evolutionary timescales. Okay, so what do I mean by sociality for viruses? Because we know what sociality means when it comes to humans or animals with behavior, right? With viruses, we're not arguing that they have behavior. The reason why they're social, I would say, comes down to two kind of fundamental processes. The two things that all viruses have to do is they have to replicate their genome, and then they have to package that genome inside virions or viral capsids to go and infect new host cells. And when you see, like in that classic image of a virus of the spiky ball, typically that's the viral capsid that's being portrayed. Now, the special thing about both of these processes is that they require gene products that in principle can be shared. So I've depicted the replicase enzyme here as a little Pac-Man. So this is an enzyme that will go around the cell and it will replicate viral genomes, provided that they have the right kind of recognition sequences at the beginning and ends of the genome. This means that in evolutionary sense, these essential viral proteins that are required for the most fundamental aspects of being a virus are evolutionary public goods. They're something that one individual contributes to the infected cell that other individuals can draw a benefit from. And we've talked or we've written previously about some of the consequences of the fact that viruses rely on these shared social processes, and there's kind of a bunch of ways that they matter. Today, I'm actually not going to talk about the ways that these can benefit the infection, although that can happen. Instead, at the risk of being sued by Disney when this goes on YouTube, I'm going to talk about the dark side of these viral social interactions at least from a viral perspective. Because anything that can be shared can also be stolen. This is true of human public goods, it's true of bacterial public goods, it's true of animal public goods, and it's very much true in viruses. In fact, it's more true in viruses than other types of organism. And the reason why is this. In order to be a virus that's benefiting from these public goods without producing, the first step is you just need a spontaneous deletion that will delete the coding part of the sequence that encodes the public good, but keeps the end bits, keeps the bits that you need to get recognized. And this is an easy kind of mutation for a lot of viruses to make. It's just a deletion, it doesn't require anything complex, any novelty, any new function, just a loss of function mutation can turn you from being a cooperator virus, producing and using, to only taking without contributing. So I'm going to use the term cheat a lot in this talk. And when I say cheat for the purposes of this talk, I'm really talking about these within cell public goods cheats. So physically, I'm talking about a reduced version of the virus that's shorter than the wild type cooperator, that's not encoding the public good, but instead is drawing a benefit. And as I've said, these cheats can arise through deletions pretty easily in a lot of viruses, and they can also get really big replication advantages. Because often within a viral infection, there'll be multiple rounds of genome replication. So if you're half the length of the other virus, that doesn't just mean you get, twice as much replication, it means you get twice as fast replication every time a replication cycle happens. So that can really accumulate to be an enormous replication advantage. So we've made the argument that these types of cheats in virology have kind of close analogies to other types of social cheating elsewhere in nature, whether that's brood parasitism with cuckoos, whether that's bacterial public goods cheats. And really, everywhere you look in the viral world, you see that virologists have kind of rediscovered entities that have this cheating lifestyle. So within humans, adeno-associated viruses are a type of cheat. Delta viruses like hepatitis D are a type of cheat. Plant viruses are full of these satellite viruses. And a lot of the entities that in bacteria help defend from phages, I would argue, are a type of kind of integrated phage cheat that lives inside the viral genome. And I like to have this figure up because it shows that although this idea of sociovirology and kind of using this explicit language is quite recent, actually a lot of the viral entities that we're talking about have been known about for a very, very long time. In fact, since the origins of the field. So the most extreme example of this, and the most relevant to this talk, is this idea of a defective interfering genome, which is one of the terms that virologists use to refer to a cheat type entity. And defective interfering genomes are a type of cheat that emerges through that process I described earlier, where you have a cooperative virus, where deletion occurs that knocks out the public goods gene but keeps the end bits. And these guys were first described in the 1940s in the very first attempts ever to grow viruses in tissue culture. We saw these kind of cyclical dynamics that are created by the kind of spontaneous emergence of these cheats that spread at the expense of the cooperator, which then goes down, as you can see that red line going down, and then into these kind of predator-prey type oscillatory dynamics. More recently, the emergence of these cheats, particularly in viruses like influenza, are an issue for vaccine producers when you want to produce a large amount of virus. And they've even been co-opted for use in biotech with things like DNA origami and phage display. So really the point is that although the language is new, the types of entities that we're discovering...
Radio now playing on Spotify.
Oh. Hey Siri, stop. Okay, let's hope Siri doesn't activate again this talk. Hey Siri, stop. Apologies.
There's nothing to stop here.
Okay, dangers of smart speakers. Hey Siri, stop. Okay, old school way is the best way. It's unplugged now. Okay, so we made the argument that these viral cheats are kind of all over the viral world, even when you're not necessarily kind of looking for them. Now, there's another reason why this idea of viral cheating has kind of come to the fore recently. And whenever I describe this process, this idea that you can get mutant viruses that kind of interfere with the wild type, one of the first kind of most obvious questions is, oh, well, that's great. Could we use this as a therapy to fight viruses? And a few years ago, I would have said, yeah, well, that's great in theory, but there's kind of limited evidence. It turns out that actually now there's quite a lot of evidence. And the way this works is like this. The idea is that you have a viral infection. In the lab, we manufacture synthetic viral cheats. Again, they're easy to make because they're just kind of reduced versions of the viral genome. We literally inject an infected animal with these cheats, and the idea is that they can replicate at the expense of the wild type, eventually suppressing or even driving the viral infection extinct. So you can think of this like phage therapy, but instead of pathogenic bacteria, it's pathogenic viruses. So like I said, this is something where in recent years, we really have an abundance of evidence for this. So there are academic labs doing this, there are CDC labs doing this, there are biotech companies doing this in a wide range of viruses, from things like HIV through to nasty emerging pathogens like Nipah virus. And I'm going to borrow here a slide that my fantastic postdoc Rowan Green put together just to illustrate the kind of timelines and kind of diversity of viral systems, where this therapy has been shown to work in vivo. So not just theoretically, but in kind of model animal infections. Okay. Are there any questions up until this point about viral sociality? Okay, excellent. So I'm going to move on to the first story now, which is going to be a little bit shorter. The final story will really be the meat of this talk. But I want to make the point in this first story that thinking about this kind of viral social interaction and kind of framing it in that language can open up new ways of answering some age-old questions within virology. And in particular, I'm going to focus on an idea called a multipartite virus. Now, a lot of us who get into biology get into it because we see crazy traits that seem otherwise difficult to explain. You know, things like birds of paradise displays or the crazy morphologies of eusocial insects. And I think for me, at least when I was first starting to learn about viruses, these multipartite viruses are an example of this. They are something that seems absolutely wild, that should not really exist, and really kind of strikes you as something in need of evolutionary explanation. The reason why is this. Most viruses look a little bit like this. You have a viral genome that produces all of the things that you need to do, replicates, and then packages together to go and infect a new cell. Multipartite viruses don't do this. They have a strategy in which all of the genes that you need are on different physical sections of the genome, all of which travel in a physically distinct virion to infect new hosts. So it's like splitting yourself apart, but all of your bits have to come together in order for a successful infection. Now, this is one of the reasons why this is a crazy lifestyle is that just kind of by design or by default, this means a lot of your infections are going to fail, right? This means that if you only get one of the parts of the genome, if you get multiple copies of the same, that can't lead to a successful infection. So then there's an inherent cost in this lifestyle. But despite that cost, these things are really common. So we've known about them for decades. They make up something like 20% of the genera that infect plant viruses, and they've evolved multiple times independently in different types of genome organization. So it's not just a one-off fluke. This is something that keeps happening. And it's not just splitting into N of 2 that happens. The most well-studied multipartite virus is farbobine necrotic stunt virus, which has 8 segments. And I probably don't have to spell out to a quantitative audience that the costs of having 8 segments that independently transmit, all trying to come together, are really going to accelerate. This is not just, 8 times harder than if you had one segment. This is super linearly, orders of magnitude more costly. So this should not exist. This should not be something that evolution keeps making. So our challenge is to explain why. Now, within the field, some of the existing ideas rested on the idea of group benefits. So there were these ideas that if you split apart your genome, maybe that allows smaller virions that survive better in the environment, or maybe that allows you to achieve different kind of regulation of your genes in different hosts. Now, I think there is actually good evidence for these ideas, but the problem with them is it's hard to imagine how they can scale in a way that scales with the costs of splitting apart your genome again and again. If being smaller means you survive longer in the environment, that's great, but it's hard to imagine how it's going to make you survive 100,000 times or 1,000,000 times better in the environment, which is the scale of benefit you need to explain these highly multipartite viruses. So I think that these ideas probably play a role in the maintenance of this trait, and they might synergize with other ideas, but it seems hard to believe that they're really driving the origins of these multipartite organisms. So the idea that I'm going to put forward is resting on this concept of cheating that I introduced earlier. So if you look at these multipartite viruses, it looks a little bit like splitting apart your genome, having one bit but not the other bit. That looks quite similar to this idea of cheating, right? This idea that you're deleting genes. So the question is, could cheating be an origin of multipartite viruses? And to spell out this idea, we're going to imagine that we have a world where we're starting only with cooperator viruses that encode 2 genes in the genome. We're going to say first, can one cheat evolve that deletes one of the 2 essential genes? Secondly, can a second type of cheat evolve that deletes the second gene but not the first? And then thirdly, can we end up in a world of only multipartite viruses? So the logic of this idea was actually first put forward by Sean Nee during his PhD with John Maynard Smith in the late 80s. And we kind of revived that idea and tried to couch it in more explicit kind of payoff matrix language more recently. So we're going to use a theoretical model here to ask a few questions of this system that will really kind of let us test the plausibility of this idea. So I'm going to start by kind of briefly walking us through the model here. So we're going to imagine a world in which initially there are 3 types of virus. You've got cooperator viruses at the top here that produce everything that they need, but they're a bit longer. You've then got 2 types of cheat. One of them produces the capsid protein, say, and the other produces the replicase. So each on its own can't successfully infect, but together they might be able to. So we're going to imagine that the cooperator virus on its own gets some amount of replication inside a cell, but either of the types of cheat on its own leads to a failed infection. And we're going to imagine that single infections occur at some rate. We're then going to say that some fraction of infected cells are infected by 2 viral genomes, which can be 2 cooperators with some replication rate. It can be a cooperator and a cheat, in which case the cooperator gets less replication than the cheat, and the cheat gets more than the cooperator, because the cheat is shorter, but it's making use of the public goods. And then we're going to say 2 of the same fail, but crucially, 2 different types of cheat meeting each other in the same cell can potentially get some amount of replication, because they're both producing the required machinery. So this leads us to a payoff matrix, which is reasonably familiar, the only difference being that we've got 2 types of defector that can potentially meet each other and complement one another. Okay, so this follows pretty simple dynamics, where we can track the relative frequencies of cheats and cooperators over time using the replicator equation. If co-infection is low, and you only have single viral genomes inside host cells, then you end up in a world where cheats can never really invade because they're always by themselves, as we expect. As you start to crank up the co-infection rate, you start to enter a world where the 2 types of cheat can invade the population. And if we ignore the possibility for complementation for a moment, we end up in this kind of situation where they eventually reach a frequency-dependent balance with the cooperator. And this is expected because the particular kind of form that we're working in here is a snowdrift game or a hawk dove game. So we expect an equilibrium to be reached where as the cheats get more common, they interact with each other more often or with the same type more often in co-infection, So that stops them taking over the population, and instead they have this balance. Now, the key thing that we introduced in our model was this possibility of complementation, this E parameter, this idea that different types of cheats in the same cell can get some replication. If we introduce just a little bit of complementation into this model, we can actually break that negative frequency dependence. We can create a world where when the two types of cheats start to become highly abundant, when they meet each other, they get some replication. So instead of the negative frequency dependence keeping the balance, we actually break it, and we can actually see the extinction of the cooperator virus. And we see in this case, this means we're seeing a transition where the viral population has been replaced by a kind of network of complementing genomes. In other words, we've seen a transition to what looks a lot like a multipartite virus. Okay, so we can see how this works kind of conceptually. The question really is this realistic? And one of the benefits of these super simple types of models is that we can extract nice, neat conditions for when we enter one world over the other. So in this case, the multipartite world exists, provided this particular condition is met, where the ingredients of this condition, these A's, C's, and betas, are to do with co-infection, they're to do with cooperator productivity alone, and they're to do with how much the cooperator pays a cost when it interacts with a cheat. So again, drawing on the fact that people have been studying these viral cheats by different names throughout virology, we can go into the literature and we can draw out estimates for these different parameter values, particularly A and C, and we can say, how plausible is this condition? If we had a multipartite virus emerging through cheats that look like cheats that we see in reality, is this a difficult condition or is this an easy condition? So that's what I'm going to plot here for poliovirus. And I'm going to plot a kind of phase space where we're going to take kind of estimates of these A and C parameters to create the space, and then we're going to plot a line in terms of E, which is complementation between cheats, and beta, which is co-infection, to see when the multipartite world exists versus when we stay with the monopartite ancestor. And this is what we see. We see that if you had cheats that have similar properties to known defective interfering genomes of poliovirus that could complement one another, assuming that we're at this red line of 1, so assuming there's no intrinsic benefit to complementation, so it's like, infection started by cheat 1 and cheat 2 has the same kind of public goods as one started by the cooperator, then you can get a transition to multipartite at a co-infection rate as low as 1/2. So if you're a virologist, this means a multiplicity of infection of about 1.7, something like that, which is well within the realm of what we consider normal. So this is a relatively easy condition, we think, or at least a plausible condition to emerge. And it's not just poliovirus we can do this for. So we can do this for a bunch of different types of viruses. And here I'm plotting different defective interfering genomes cheats. So different viruses where people have gone out and measured the emergence of cheats that delete a public goods gene, measured how much interferes with the cooperator virus, and we can kind of plot here the phase that we're in. So in principle, this seems to be a relatively plausible explanation based on the known properties of cheats. So I mentioned these collective benefits arguments in the literature. So the next thing we can do in this model is we draw a line between on this kind of red dotted line, which is separating the world where cheat infected cells above it, they're doing better than cooperator infected cells. So that might be if the smaller virions can survive a bit longer, you might be above the line. Whereas on the bottom, you've actually got an intrinsic population level cost to being multipartite that we're putting into the model because we're assuming that splitting apart your genes is, for whatever reason, less good than keeping them all together. So you can see that in all those examples, we had this blue space in the bottom right of the plane where multipartite is invading, even though cheat-infected cells are intrinsically worse than cooperator-infected cells. So we can actually ask, is this a general condition? Does this always exist? And it turns out it's true. So we can take the population level mean fitness for the cheat equilibrium, compare it to the cooperator equilibrium, and we can actually show that this space always exists. It's always possible for multipartite to invade in a way that the resulting population has a lower mean fitness than the ancestral population. So strictly speaking, group benefits are not required for cheating to work as a mechanism for the origins. Okay, so I mentioned that one of the real problems with multipartite are these highly segmented viruses where you've got, more, not just two, but many. And the problem was that the collective benefits arguments don't really scale as you split further and further. But if we don't need collective benefits with cheating, could this provide an answer to this kind of many segments problem? So to tackle this, we moved away from the nice analytical model and we moved into a slightly more complicated agent-based simulation, where what we're doing is we're simulating a viral genome that in this case has 8 potential public goods, each of which could be cheated. And we're assuming you can get cheats and kind of any combination emerge. So maybe they delete genes 2 and 3, but they keep the rest. Or maybe they delete gene 5, but they keep all of the others. And here we're on the E equals 1 world. So we're assuming that there's no intrinsic group benefit to multipartite. And what I'm going to show you here is stacked bar charts where we've run 100,000 or so simulations at different multiplicities of infection, which is controlling the co-infection rate. So higher means more co-infection. And I'm going to show you the fraction of simulation runs in which the resulting population has different splits to different multipartite worlds. And what we find is that as you start to crank up the co-infection rate, even keeping it relatively modest, so below 10, you can start to get splits to highly multipartite genomes with as many as 4 segments. So what's happening here is we're blowing up the parameter space within which we can explain the evolution of multipartitism. Instead of being constrained just as n equals 2 problem, we're potentially explaining kind of a much wider range of parameter space because we're breaking the assumption that the resulting population needs to be more fit than the ancestor. Okay, now the final part of the story is this is nice, but really what you want to do is you want to test the predictions. You want to see, okay, not just could this happen, but has this happened? Is there evidence that this might have happened in reality? Now, it's difficult to do this kind of thing in viruses because we don't have we've got such a tiny sampling of the virosphere, and we don't really have well-measured traits for a lot of viruses that we're interested in. So this is my attempt to do this, which is looking at the 5 of the what we believe to be independent evolutionary origins of viruses. And we're kind of seeing that overall genera for which people have measured, or rather origins for which people have measured a greater number of viral genera that are known to produce cheats do seem to also be those origins that have more multipartite viruses. But this is not enormously compelling. It's not a strong test of the idea, which leads me on to the second part of the talk, where I think we can have better answers to this. But it's at least consistent with the hypothesis. So just to sum up the first story, we've made the argument that cheats can drive multipartitism under fairly easy conditions, without needing collective benefits, in a way that can produce wholly multipartite viruses, in a way that's at least consistent with the empirical patterns. Okay, do we have any questions at that point on the first story about multipartite viruses? Okay, great. In which case, I'm going to move on to... Oh, we've got something in the chat here.
Yeah, question about the previous slide. Yeah.
Yes, here's the previous slide. And this work's published, by the way, a couple of years ago. So more details are in the paper.
Was there a question about this slide? Oh, okay. Just to see the slide. Okay.
Cool.
Okay. Yeah. No specific question. All right.
Perfect. Okay.
Right. Carry on. Thanks.
So I'm now going to move on to the next one, where we're going to move away from the evolutionary timescale into the within host timescale. Thinking not about millions of years, but rather about days. Now, the reason why this cheating idea potentially provides a plausible mechanism for things like multipartitism, is that we've argued that it's like an intrinsic property of viruses that, happens a lot, and we see examples of cheat species all over the viral world. Which is true, but there's a caveat to this, and the caveat is a lot of our knowledge of viral cheating actually comes from laboratory studies. In particular, the argument that cooperator viruses can generate cheats, which was part of our multipartite model, this is something that we see happening in the lab a lot. This happens to most animal and plant viruses when you grow them in high co-infection conditions. But, right from the 1940s. But it's an open question whether this also happens in natural infections. Because for a large amount of the time that we studied viruses, it wasn't really possible to get any insight into this, right? Like you need sequencing data in order to start to peel apart the viral population. So there's this question that I think occurs to us naturally, not just from a virology perspective, but also from an evolutionary perspective. What is the natural history of these things? Is this idea of cheat generation just a lab artifact, or is this something that is really mattering in the natural ecology of viruses? Now, there's a couple of things that are helpful here. One thing is that because the most common type of viral cheat here is this defective interfering genome that's deleted the public goods genes, this makes it relatively straightforward to search for within genome sequencing data. And to show you how we do this, I'm going to walk you through the background a little bit with influenza virus, which is the virus that I'm going to focus on for the next part of the talk, because we have lots of sequences from it because of the unfortunate outbreaks that are happening at the moment. So influenza has a genome that is segmented rather than multipartite. So it's essentially got 8 chromosomes, each of which encodes a different gene. 5 of these encode genes that we think are not easily shared with other genomes, or at least they can be, but not in an exploitable way. 3 of the genes instead encode the polymerase complex, which is this replicase, it's a little Pac-Man there. And so here I've got the different colors to indicate that the Pac-Man for flu is actually made up through 3 different subunits. And at least in the laboratory, deletions on these 3 polymerase segments are the ones that become cheats. These are the ones over on the population. So we've got a kind of target to look for. Okay, so the way that we can find them, so a great collaborator of mine, Andrew Routh, who is now at Scripps and ClickSeek is his company, has an algorithm that is a little bit like a kind of splicer-aware aligner for eukaryotes. We go through all of the reads from the viral population, and we're looking for specific reads that look like that cheat read at the bottom there. Individual reads that have got a big gap in the alignment, which indicates that that read comes from a deleted viral genome. So we're not using differences in coverage to infer these. We're actually using specific reads that map to what we think is a cheat genome. And this is an approach that works very nicely on lab data where we know that we get lots of cheats. And just a note on terminology, if you come from the virology world, defective interfering genomes are a type of cheat. Defective interfering genomes are a subset of defective viral genomes. Not all big deletions act as cheats. So here we're focusing on the big ones of the polymerase complexes that we know have evidence act as cheats from the lab. So again, just to kind of give an indication what we're looking at here, what this looks like when you kind of visualize it, the input data for most of what I'm going to show you are the abundances of these cheats that we're inferring from reads that span these sections of the genome, indicating that there was a viral molecule that was at high abundance that deleted part of the genome. And we're going to use this approach within the H5N1 avian influenza outbreak, which you might have heard about because it's all over the US in particular, kind of agricultural animals like cows and chickens and things. But this is also a virus with a very broad host range. So here we've got things you expect, like birds and mammals, but you've also got some really exotic mammals, like polar bears get this, sea lions famously get this. So this means that we have a super broad range, both of viral genotypes that have kind of emerged over the last couple of years and different host species to look at. So within our data set, we've got about 10,000 samples overall split across these genotypes and host taxa. And Andrew, together with Praneeth, put together an impressive computational pipeline that means that we can sample all of these kind of sequences that have been put up from this outbreak, look within the kind of deep sequencing file, apply Andrew's algorithm, and get an output that kind of breaks down the viral population within each sample. And there's two assumptions about the data that are important. One is we assume that, so these samples have been taken from animals. We assume that the sequencing on that sample is broadly reflexive of the viral population within that host. So they might be taken from a particular, part of the body or a particular tissue, but we're kind of assuming that it's a good reflection. And the second really big assumption that's important for how we interpret the upcoming figures is we're assuming, based on the small bottleneck size that people know about influenza, we're assuming that most of these infections were started by just one or a small number of fully functional cooperative viral genomes. So we're assuming that any cheats that we detect are emerging kind of within that host infection, that they weren't there at the beginning. Okay, now the second part of this talk is really leaning very heavily on work that Rowan, a fantastic postdoc in my lab, has done. And he's done this really rapidly, really over the last 6 months or so. So the first thing I'm going to show you is I'm going to show you data where we've taken all of the sequencing across all of these 10,000 infected animals. We've pulled the whole data set, and we're detecting viral genomes that have deletions that are greater than 30 nucleotides. And we're doing this because we're interested in these deletion-containing cheats. And we think that if you're deleting more than 30 nucleotides of the coding sequence, you're definitely not encoding like a functional normal version of that gene product. So it's kind of a conservative way of estimating a defective genome. And what I'm going to plot here is over this whole data set, just bulk, every deletion event kind of binned into different areas, depending on where that deletion started and ended. And we're going to do this first for the non-structural protein, which is one of these non-shareable proteins for flu. And this is frequency data. So this is how often is these deletions cropping up. And so what you can see are, so anything along this kind of white line, those white squares, like you can't have, we're filtering to more than 30 nucleotides. So anything roughly along that y = x line is kind of shorter deletions where you're kind of ending close to where you start. Anything in the top left is where you're kind of starting early, where deletion kind of starts early in the genome and then continues later. So that would indicate a large fraction of the genome being deleted. And you can see that for this kind of virus, we're getting this, kind of a reasonable number of these deletion events occurring, but there's not a particularly obvious clustering. They're kind of roughly kind of splashed all over the plot. Now I'm going to contrast this with when we do the same plot, but for deletions that we've detected in the polymerase gene, which is one of the ones that we know contains these big deletion cheats. And I want you to focus on this bright red spot in the top left corner. What you've got here is a really strong clustering where we're getting a very large number of deletion events that are large, that are starting near the beginning of the genome and ending near the end of the genome, indicating that you've got a deletion that spans pretty much the entire coding sequence. And you can see this really striking clustering. So yes, you've got some along that y = x line, but almost all of the genomes that you're counting, and also just to point out, this is a log scale. So this is kind of underrepresenting the real scale here. There's a real clustering in this top left corner of the genome. So over the whole data set, this pattern repeats for the polymerase segments, not less so obvious for the non-social ones. And overall, we're getting nearly 1 million unique deletion-containing kind of cheat-similar reads across this data set. Now, when we break this down by abundance, the pattern becomes even more striking. So on the left here, we've taken all of these deletion reads, and instead of just mapping their frequency, we're weighting them by our estimate of their relative abundance within the host. And we can see that across this whole data set, more than 90% of all of the reads that map to deletions are mapping to large deletions of the polymerase segments, that top left-hand corner of the plot. So it's not just the highly common, but they're also reaching super high abundances. They're really dominating the within viral population, within host viral population. Okay, so the next thing we can do with this is we can say, okay, so overall, these cheats or these genomes that share the hallmarks of cheats are reaching high abundances. What about if we break this down on a host by host level? So what you're going to see here is on the x-axis, we can kind of plot in its percentile individual hosts in our dataset across the 10,000. And on the y, we're going to plot the estimate for the fraction, our best estimate for the fraction of deleted viral genomes kind of within that population, within the host. And two things I want you to focus on. The first thing, on the left, basically everybody has cheats at some abundance. So if you set our DVG abundance cutoff at like 1% or so, 95% plus of samples in this data set have it. So if you compare this to like minor variants where 1% might be a cutoff, everyone has them. But the really striking thing in this database is this intersect here. What you're looking at here, the vertical kind of line kind of going horizontally separates the space where cheats are making up 50% of the viral population within the host. We find overall across these 10,000 infections, about 1 in 3 individuals have cheats, not just there, but at more than 50% relative abundance. In other words, the majority variant. And bear in mind that these cheats are non-functional in the typical sense. They can't be producing a normal version of the protein. They're defective. They're obligate. Nevertheless, they're making up more than half the population a third of the time. And we can then break this down by different genotypes and by different host species. So here's a comparison where you've got landfowl, which is chickens and turkeys and things, infected by two different genotypes, B313 and BD1.3 and D1.3, different genotypes of influenza. The fraction that are cheat dominated is varying by about 5-fold. So this is an enormous effect of the cheat genotype on the resulting population. We can also do this with different host groups. So here you've got D1.1, which is the lineage that we're really scared about because it infects like a super broad host range, which allows for these comparisons. So waterfowl, so ducks and things, and landfowl, so chickens and things, similar kind of intercept here, right? About a third or a bit higher. Hawks, more than 2/3. So not just genotype is mattering, but within the genotype, different host species are cheat dominated at different extents. Now, that's a kind of visual description, but Rowan went further and put this into a really nice kind of GLM, where we're doing a beta regression modeling the cheat abundance in these different host and genotype combinations. We find a very strong interaction effect and a reasonably good R-squared for kind of ecological kind of data. And this is kind of what it looks like in these different groups. So you can see that the predicted abundance is kind of a property of both the genotype and the host. And this allows us to kind of make some generalities. So one is that B313 lineage, which is the one that's mostly in cows, is more DVG prone than D1.1, at least for the comparisons that we're able to make overall, and actually reasonably significant in the comparisons for which we have a lot of data. On a host level, we can also draw out these generalities. So cats, canids, and skunks and stink badgers have much, much higher DVG loads on average than other groups for both genotypes. The opposite seems to be true for things like waterfowl and things like hoofed ruminants. Again, reasonably strong effect sizes across multiple comparisons here. There seem to be these host and genotype specific biases that are coming out of the data. Okay, that's true for cheat abundances, so there's the total amount of cheats in the population. But what about the specific cheats making up that population? So Andrew had a clever idea here to draw an analogy to single-cell genome sequencing. So what we're doing here is we're going to say, imagine each infected kind of host, each kind of host sample is like a single cell. Imagine we've sequenced it, and imagine the flu genome is kind of expressing these DVGs at different abundances and different specific DVGs. And this allows us to use UMAP, which you might have seen before, which is kind of like a fancy principal component analysis. And the first thing you can see is we just throw all the data into this and we focus on one of these polymerase segments. It's a very clear vertical separation where different genotypes are separating in different areas of the space. So we're feeding this just the cheat population and the abundances, and it can start to orient you. It can say, okay, you're a one genotype versus another. Now, the important thing about this is it's not just caused by that cheat abundance effect we saw earlier, because cheat abundance is varying on a different axis. So there's something other than cheat abundance that UMAP is able to use in order to separate these different genotypes. And this is true not just for genotype, but also, this is true firstly across all of the polymerase segments in particular, not just for genotype, but also for host species. And this is kind of bulk, but this is true when we break this down. If we look just within a genotype, we can start to separate the different host species that genotype might have come from purely based on the different cheat populations in this space. Now, this is very much kind of a preliminary finding here. Andrew has gone one step further into this, and he started to pick out specific cheat sequences that are overrepresented in some or the others. We think that this might allow you to turn the host population into the sheep population into a kind of signature of the host that you've come from based on these sequences that you do or don't have in an environmental sample, for example. But it is somewhat preliminary. So the final part of this kind of data story. So I mentioned host groups earlier and how different species and different genotypes have different sheep populations. You might have been thinking, what about humans? Humans are a host group. So the human data that we have is not H5, it's seasonal H3 and H1 influenza that comes from Kirsten St. George at the Wadsworth Center, New York State Department of Health, who's been very generous and shared a couple of hundred sequences with us. And the headline is this: for humans, for seasonal influenza, the point at which the fraction of the population where the population is cheat dominated, we put to be about 2/3. So this puts us on the upper range of these host species we were looking at for H5. At least for the human populations that we're sampling, there's a 2 in 3 chance that cheats are the dominant variant, more common than the wild type within the population, which is high. And so the next steps of this project is Rowan's going to start to be kind of really getting into this, testing if this cheat abundance could potentially be a biomarker to predict the severity of the infection. Okay, so I'm going to sum up this part of the talk, and we're getting close to the hour mark. So overall, kind of going away from this world of theory and more towards this world of what's going on in real infections, across a very large number of samples, basically every infected animal had detectable cheats, or at least genomes that look like cheats for influenza. These were by far the kind of dominant form of within host viral diversity within these influenza infections. And in a third of cases, they dominated the infection in a way that varied depending on the genotype and on the host taxonomy. And for humans, we're in the 2/3 part of the range. Okay, so I think that overall, The story here is that these viral cheats are not just a lab artifact, but as best we can tell, they're a really substantial component of within host viral evolution for influenza. So I'm going to very quickly go through kind of where I think this goes next. So this is a striking observation, right? This is a puzzling observation in the sense that if the majority population is a cheat genome, that seems like it should be an evolutionary cost. So what's going on here? There are different ideas about why there might be so many. My argument that I'm going to put forward to you very briefly is I think that it could just be the case that these viral cheats are a kind of emergence of evolutionary conflict within a population, analogous to things like cancer and multicellular organisms. And this is actually an idea that virologists have kind of been talking about for a while. It might be the case that it's not just cheat therapy that could end an infection. It might be the case that the generation of these cheats and then them spreading and taking over the infection is a process a little bit like immune responses that is just like a natural limit on the rate at which viral infections spread. So there are a few ways of kind of attacking this problem, looking at traits which I kind of don't necessarily have time to go into. But one way of thinking about it is breaking down this first part, the kind of generation of cheats as a function of the flu genome, whereas this replication of the cheats is a kind of co-evolutionary function of both genomes. So in this data set, for example, we find there are specific motifs, such as having more uracils, that appears to make it more likely that a cheat is generated. And Rowan has some awesome ideas about how we could use those kinds of motifs to start to tease apart mutation from selection in kind of studying, is this cheat generation an evolvable phenotype of viruses? And then the other angle is to really start to get into modeling these cheat dynamics once they've arrived, because really this headline figure of 2/3 cheat dominated is a property both of the virus and of the cheat. So hopefully I've been able to make a case that is at least interesting to think about viruses being social. And this can provide a new way of looking at viruses. So the next time you open a textbook and see a virus, I'm hoping you don't just think of a solitary particle, but it's a complex community that in fact might be, in its natural state, majority dominated by cheats. So with that, I'd like to thank various collaborators who have enabled this work, especially Andrew and Kirsten for the flu data. And really most of the work, particularly in the flu side of this talk, has been done by Rowan, a postdoc in my lab. Now, Jesse mentioned I've started recently in Canada, which is true. And we've been developing this kind of socio-virology idea through having conferences where we get like a super broad range of virologists and evolutionary biologists. And I had this idea kind of as soon as I knew I was coming here, that it would be nice to bring this community to Canada. And I am delighted to announce that we have now done this. So this August, we're having a meeting in Vancouver on UBC campus. Accommodation and registration and food are all covered by a grant from CIHR. And it would be great to see a super broad range of people. So get in touch if you're interested in this. Thank you.
Fantastic. Thank you, Asher. Very exciting stuff. Do we have some time for questions? So feel free to speak up or put something in the chat. Yes, a question from James.
Go ahead, James, you're muted currently.
Sorry, okay.
Yeah, we can hear you.
Can you turn that off?
Unmute this one.
Yeah. Okay. Thank you for your talk. That was very interesting. I admit that is my first knowledge about viruses, so whatever I know is what I was able to gather from what you were saying. But I'm curious about the fidelity machinery viruses have during replication. Like, would you expect, since I imagine they're a little bit simpler and they're error correction machinery, that you would expect for these kind of cheat partial genomes to arise just through stalled replication? And is that kind of change, like this maybe would be an expected thing to happen without any kind of evolutionary driver?
Yes, great. So yeah, so the question is to do with the mechanism of how these form, and is it kind of inevitable? So yes, I think it is inevitable. Part of the reason I think it's inevitable is there's some fantastic work from labs that look at RNA replicator systems where you only have kind of the genome being encoded, and they also see emergence of similar kinds of cheats. So I do think the formation of these things is probably not something that viruses can fully escape. However, it is also true that there are specific motifs that can make the likelihood of that error occurring more or less likely. So we know that uracil-rich tracks make polymerase stutter. We know that distal homology of mutated genome can make it more or less likely. So there are evolvable traits of the genome that could affect formation. Now, I think the kind of second part of this is, do you need selection? So that's talking about selection on the part of the cooperated genome in terms of producing them. I would argue that you don't need anything clever to explain why these things are kind of generated. You do need selection on these cheats to explain why they're achieving more than 50% of the relative abundance. Like these things are not forming randomly, or rather, the highly abundant ones are not found randomly throughout the genome. They're more than 90% coming from more than 3/4 deletions of the polymerase genes. So I think you need in order to explain why these things are so abundant, you need to invoke the idea that they are exploiting the wild type genome to reach a high relative frequency.
Other questions from the room or I've got a question if I may. Oh, or did someone just go up to the
Yeah, I had a question. So, regarding the evolutionary advantage of having cheats, doesn't it make sense to like not have all your eggs in one basket? So it's like harder to wipe out a particular multipartite viral system as opposed to just as like a monopartite virus?
Yes, okay, so I guess two things. So one is you might imagine that working. Okay, so firstly, I'm going to say that I don't think that works for the influenza cheats because the influenza cheats just like fully lack like the that coding sequence. So from a kind of population level, that cheat can never recapitulate an infection. So it doesn't matter if that cheat survives from the kind of perspective of the rest of the virus. It doesn't matter because that can't reinitiate it. But that might be different from multipartite virus, or as you say, maybe having it across different virions is allows you to kind of survive. Possibly, the argument that I always come back to is these kinds of arguments. It's easy to imagine why that might give you an advantage. It's hard to imagine why that gives you a 3, 4 order of magnitude advantage, which is what you need to explain just the viruses that split to 3 or 4 segments, let alone the ones that go 5, 6 or higher. So that could be a potential benefit, and I think those benefits are probably important in why these multipartite lineages persist over evolutionary time. But in terms of the origins of it, I think you need something that doesn't rely on a group advantage. Thanks for your question.
other questions? I do have one or two. I know we're past the hour, so I'm sensitive to if folks need to go. But Asher, if you have like
Okay, by me.
no strong constraints. Okay, well then I'll if you'll so yeah, super interesting stuff. I'm curious if the host species effect for the influenza and the first part of the talk, you talked a lot about multiplicity of infection. Is that something that could differ across host species? And might you have met, like, I don't know if we have some estimates of this or even like viral load by qPCR, like, yeah,
Mhm.
could it be explained by MOI and might you have the data to look at that?
Yes. Okay. So it would be amazing if the co-infection rate did not vary among these host groups, right? Like that, and then there are different components to this. So that initial model was super simple, kind of well mixed population, MOI is constant. Obviously, within a host, you've got different tissues. You've got potentially a different co-infection rate in each tissue. You've potentially got like immune responses and making like a different dynamic. So there's a lot that might be happening. So I'm absolutely certain that that is part of the answer. I think the hard part is trying to kind of map on what about the host might be leading to a different MOI. I like your idea of just using some bulk measure like total viral load, like like Alison Fader's got some great work showing that higher viral load is associated with higher recombination rates, so presumably higher co-infection. With sheets in particular, there's a little bit of cause and effect thing going on there, because once the sheets start spreading, you then expect that to decrease the viral load. So it's not immediately obvious what you would predict, which is why for the clinical effect, Rowan's got that nice causal path diagram where we're going to try and tease it apart in a little more detail. Now, the other kind of caveat or the other complication to that host data is the data was collected in different ways. So some of our like skunks and hawks and that kind of thing, the data we think is from hunting, catch, where they've then sequenced it. Whereas for stuff like the chickens or stuff like the cows, that's more from farm surveillance.
Yeah.
And even within that farm surveillance data, there's going to be different protocols, which mean that we're probably catching the infection at different points for these different species.
Yeah.
So one kind of null model for this, which is kind of what I was starting to look at with some of the later modeling, is maybe it's exactly the same process that plays out in every single host, and we just catch them at different points, and it's just a kind of dynamic process.
Right. Yeah. Makes sense. Any last questions? All right. Well, fascinating stuff. Asher, thanks for sharing, especially the latest unpublished results. Very, very cool, and excited to see what comes next. And I encourage everyone who's interested to check out the social viruses meeting in the summer. And yeah, there's the ad again. And thanks again.
Perfect. Thank you so much, Jesse, and thanks everyone for coming.
Thanks a lot. Bye bye.